Namaste guys, this is Sachin from Alliance University creating a informative video on confocal laser scanning microscopy. Beside this topic, it's my art channel and I'm uploading this informative video because due to an assignment from our physics professor Vipin Prasad sir, and I am thankful for the sir to giving an opportunity to present something different from art. So let's start uh, the subject of confocal laser scanning microscopy. Introduction. Confocal laser scanning microscopy is a powerful imaging technique which in which we can get the image at a high resolution, which will give you a clear view of that image. We have come through many microscopes in our past days. Why so we called confocal laser scanning microscopy as a powerful imaging technique? Yes, this is due to its image at a high resolution. The image it provides will be at a high resolution, which will give more clear concepts on the structures, which will help to the scientists, researchers, almost all. And apart from this, it will give you a three-dimensional image also. Three-dimensional image will also help for scientists to observe the observe the tissues, structures, cells clearly, and get an idea. And there's arise a question that how can it generate high resolution three-dimensional image? How could it? And how it is different from other microscopes? It uses just two components, a laser beam and a pinhole aperture. A laser beam and a pinhole aperture. First one, the laser beam will just to scan the sample. It will scan the sample fully, and we use a pinhole aperture, which is to be a function which is used as a it just allow the focused light and directly block the non-focused light. Means we can understand that the high resolution image of that thing is mainly depend upon pinhole aperture because it blocks off focus light and also allow only focused light. So we can conclude that the laser scanning microscopy is give a high resolution image. And what are the uses of this confocal laser scanning microscopy? It used in various fields like biology, material science, engineering, study of cellular structures, tissue organizations, surface morphology, many fields. I will give you an example that we have learned many things in our class 12 in biology, and that we have learned the biological tissues, and we have seen many pictures in our textbooks about the biological tissues, how they are formed, how could be they are many clearly, because we can't see biological tissues in our naked eyes. So uh, what is the main point of this CLSM? Its main point is focus. The elders have created a quote that just focus on your work, just focus on your work, the success will be in your hand. Focus on your work and the success will be in your hand. Like that, the focus of confocal laser scanning microscope is that much strong that it focus on a specific region in a perfect region, and uh, we can change the region to know more knowledge about the thing, biological resources, tissues, cells. So its use of CLSM could be in various fields. With that, why it is useful? Because of its focus on specific regions, which helps for the scientist to observe the sample clearly. And what are the components used in CLSM? Laser source, scanning mirrors, pinhole apertures, detector. Laser source for scanning the sample, scanning mirrors which directs the laser sources, pinhole aperture which is the main component because it only allows the focused light and blocks the non-focal light. Detector, which detects the wavelength of the light wavelength, and also give a perfect picturized image. These are the components combined to form CLSM, confocal laser scanning microscopy. Move on to next. Process. What are the process? I will tell in a simple way that first the microscope will emit a laser which will be directed by scanning mirrors and which enters to the across the sample, where the sample emits the light with a different wavelengths. That type of different emitted wavelengths light is collected by the detector. The detector, which is present over CLSM, will collect the emitted light by through pinhole aperture, which only allows the focused light to pass through. After that, we are at the conclusion that CLSM, confocal laser scanning microscopy, is really a valuable tool. Is it? Yes, of course, because it gives a more high resolution image. With that, also gives a three-dimensional view of that particular thing, substance is like a non-imaginable thing. So it is really a valuable tool for the scientists, researchers to get a clear view about that particular thing, substance. And also, uh, I am concluding this video over here because I have given uh, time interval has just ended. Okay, guys, I hope the concept is clear for you. And if you have any doubts, you can ask me in the comment box. I will try to answer it. Uh, do subscribe. Thank you, guys. Thank you.